My name is Willow, and today I'm going to be talking about the experiments conducted by Nierenberg, Mattei, and Leder. They deal with the topic of translation and specifically codons and amino acids. We learned in class that a codon consists of three MNMR bases that form one unit. In 1961, Brenner and Crick established that a codon consisted of three non-overlapping nucleotides. We know that humans only code for 20 amino acids and need 64 codons to do this. Now that scientists had figured out that a three-letter combination coded for an amino acid, the next question was, which three-letter code pairs with which amino acid? In 1961, the first experiment to take place was done by Nierenberg and Mattei, with their main question being what amino acids specified by the codons composed of only one type of base. For example, what amino acids go with UUU or AAA? To solve this, they first took a poly-U MNR chain that consists of tons of U's put together. They then created a cell-free extract which contains all the components of a cell, but they are not confined to the cell, allowing the poly-U chain to be able to access the cell's nitrogenous bases. Once these two parts were mixed together, they then added all 20 of the amino acids to the solution, with one of them being radioactive. The radioactive element was crucial because it allowed the specific amino acid to be identified. They repeated the experiment 20 times, and each time they used a different radioactive amino acid. They found that the only time they could get any sort of radioactive reading was in the tube for phenylalanine, meaning the only time they formed a protein successfully was when the radioactive amino acid was phenylalanine. This proved that the codon UUU encodes for phenylalanine. Using the same experimental setup, they went on to find that CCC encodes for proline and AAA for lysine. Nothing encoded for GGG. Through this work, they were able to find what amino acids 3 of our 64 codons coded for. A few years later, in 1964, Nierenberg and Leder did a follow-up on this experiment to determine what other matches between codons and amino acids could be made. They took very short pieces of mRNA, in my example, GAG, tRNAs with amino acids and ribosomes, and mixed them all together. They did the experiment several times using different radioactive amino acids. They then filtered out the unbound amino acids and looked to see if the filter paper was radioactive. If the tRNA with the radioactive amino acid did bind together, they would be caught by the filter paper, and this would prove that the radioactive amino acid corresponded to the mRNA determined at the start. In my written out example, this proved that the codon GAG encoded for the amino acid glutamic acid. They were able to use this experiment to identify the remainder of the 61 codons, which led to the creation of the coding dictionary. This experiment was important because the language of DNA and our genetic code now had a universal format to be understood and transcribed.